Welcome back, ladies and gentlemen. So today we're going to continue our discussion on microevolution, genetic evolution, um, and since the whole idea of microevolution is to be able to look at frequencies of characteristics and how they change, that means we can actually calculate that from one generation to the next. And so that's what we're going to kind of take a look at today. So first off, we've got to get some basic vocabulary here. And that's this idea of a population's gene pool. So when we talk about the gene pool, we're talking about all the combined alleles of all the individuals in a population. And we can drill into that gene pool and look specifically at the combination of traits for a particular characteristic, or we can talk about the gene pool collectively for all the traits that exist in that population. So most of the time when we're looking at microevolution, though, we want to drill into a specific characteristic, hair color, eye color, fur color, whatever the case might be. And so I want to look at both the allele frequencies as well as the phenotypic frequencies. So remember your genetics terms here, right? When we talk about an allele, we're talking about a specific characteristic and the different variations that exist for that characteristic. So when we look at traits like that, then we're looking for your essentially your letters, your capital A's, lowercase a's, that kind of idea. Right, so when we're looking at those allele frequencies, we're really talking about the ability here for something like your capital A, right, versus your lowercase a. So what's the frequency of being dominant versus what's the frequency for being recessive? When we come down to your phenotypic frequencies, however, we're now looking at how common is a particular phenotype within the population. So if I know that being dominant, right, which would be that capital A, or somebody who's heterozygous for it, is brown, then I'm looking for the phenotype of being brown, or I'm looking for the recessive, right? In which case their genotype would look like this, right? And maybe that's white. So that's the idea here. So we're looking for the frequencies either of just the capital A allele, the capital or the lowercase allele, the A, because every individual has to carry two of these, then we've got to calculate how many have two of this, how many have two of this, or how many have one of each. That's the idea here for your allele frequencies. When I'm looking at phenotypic frequencies, I now just want to know, well, how many are brown versus how many are white? So let's take a minute and actually try this out. So I've got in a population of mice, black eye color, all right, which is going to be our dominant trait, and that's dominant over the blue eye color, which we'll use as the lowercase. So I've got some diagrams over there to help us try and answer the questions. I've got my sample population in a pie chart up top, as well as the gene pool sort of graphically laid out for you, where those really dark circles are your capital alleles, your dominant alleles, the sort of more gray circles are the lowercase ones. So you've got three questions here to answer. First off, in the gene pool, how many alleles are accounting for the black, the blue versus the total? What's then the allele frequencies for your dominant versus recessives? And then what is the phenotypic frequency for those two different colors, the black and the blue? So take a look back on the previous slide and go through those equations and see if you can answer the questions based on the data that's sitting here for you. Okay, so let's take a look at this. So first and foremost, in my gene pool, I have to know how many alleles there are of each color. So how many have that big B versus that little b allele? And you really have two ways based on the information that's here to go trying to tackle that question. If you didn't remember what heterozygous, homozygous meant, you could just go down and count the circles in that gene pool diagram. But I want to take us a step back and let's go back up to what these terms actually mean. So if I'm going to look at somebody who is heterozygous, right, that means that they have one of each. So that would mean they've got a big B and a little b, right? If I'm homozygous for being black, remember black is the capital, so that means these guys have two big B's, right? And if I'm somebody who is homozygous blue, that means that I've got two lowercase letters, right? So when I look at the ability for the homozygous recessives to contribute, that really means that they're contributing two of them. So there's 9 individuals here, but they're contributing 2 of those lowercase b's, so total of 18 lowercase b's that they're contributing. If I look here, these guys are dominant, so there's 4 of them, right, but they are contributing 2 each, so I have the total of 8 big B's from there. These guys up here are each contributing individually 1 of each, so 12 big B's and 12 little b's, and then I'm going to add those guys together. So ultimately what you end up seeing is if you add 12 plus 8, you get 20 of those blacks, which remember are the big B's, right? I add those blues together, the 18 from those 9 individuals plus the 12 from the other, and I end up with 30 blue, which again would be those lowercase b's. And so ultimately I have a total of 50 alleles in my population collectively for this given eye color trait. Okay, so what does that then mean for the allele frequency? Well, when I look at allele frequencies, right, now I want to know what's the percentage, really, of the population that has one trait versus the other out of the total collective available alleles in the population as a whole. So if I take a look at this, that then means that those big B's, we had 20 in that original population out of 50. And so I have a 0.4 frequency, which is often how they end up leaving these when we calculate this, by the way. But to just give it to you in a way that maybe is a little more familiar to you, it's 40% of the population. I do the same thing with those lowercase alleles. For blue, there were 30 out of the 50 total, and so 60%. Okay, now I want to come back and take a look at the phenotypic frequency. So how common is it to just have one color versus the other? And again, out of the total population. So now here's where it's helpful to really go back to that pie chart again. If I go back up here, right, I've got 12 individuals who are black, plus 4 individuals who are black, 9 individuals who are blue, and if I were to add that up total, how many individuals do I have for the entire population? So that means I've got 16 individuals who are black. There are 25 total, and so I have a 64% frequency for being that black color. 9 individuals out of the 25 who are blue, and so I end up with 36%. So what does it ultimately mean to have those numbers available to us? Well, what it allows us to do is to create a situation of predicting what allele frequencies should be if nothing's actually going to happen, meaning that there's no natural selection, there's nothing that's changing those genes, because if they just go through typical sexual reproduction, it's a very predictable pattern. Those frequencies for the entire population, so long as nothing is happening to that population, stay very consistent. When we go to make those sex cells via meiosis, which we'll talk more about later this year, and then how those gametes, those eggs and those sperm, come back together during fertilization to make the offspring, it's very much separate, put back together, separate, put back together. And so you're never really changing what's available when you look at the population level, unless something else is happening. And that's this idea of Hardy-Weinberg equilibrium. It simply states that your gene pool remains constant for the frequency of alleles that are present at the time and for every generation thereafter, assuming that nothing is acting on those genes except for the creation of sex cells and the recombination, bringing those sex cells back together to make an offspring. Here's the thing. Hardy-Weinberg equilibrium rarely exists in nature because there really is no such thing as a population that isn't being affected by natural selection or other environmental factors. So Hardy-Weinberg isn't necessarily something that really occurs in nature, at least not commonly, but it does provide us a really nice no change baseline to compare to. So if we just look at a population set of genes and say if nothing happens, this is what it should do, this is what it should remain, then we can look at successive generations from there and see if anything is in fact changing in those gene pools and be able to start discussing what might be causing the changes if we see them. So here's the thing. There are 5 conditions that need to be in place in order for there to be this non-evolving population. And again, most populations will fail one or more of these when we look at them truly functioning in nature. First and foremost, they have to be extremely large population sizes. So the smaller the population gets, the greater there's a chance of just random fluctuation in their allele frequencies from one generation to the next. Something we'll talk a little later about as genetic drift. The smaller the population, what really ends up happening in the sense of genetic drift is a handful of things, but what we're drilling down to is the idea of inbreeding. So the smaller the population, the more likely you are mating with somebody who is more closely related to you than you would in a much larger population, and as a result, characteristics that might actually be harmful to the population actually become more common than they should, simply because there's really no other choice. Um, and so the bigger the population size, the less likely we have for those sorts of fluctuations to occur at random. And so any fluctuation we see is more likely a result of things like natural selection, choosing, quote unquote, a trait because it's beneficial. We also can't have any gene flow. So remember, I need to keep my population stable, and gene flow is the transfer of alleles between populations, meaning that somebody's either immigrating or emigrating from that population, right? So immigrate means to move in, emigrate means to move out. And if I have individuals moving in or out of that population, they're taking their alleles with them. They're taking their genetics with them. So if somebody moves in, that means they're adding to that population, and that's going to change the frequencies. If I have somebody leave, the same thing. They'll take those alleles with them and again change the allele frequency. The whole point of Hardy-Weinberg is that nothing is supposed to be changing. It's supposed to be a no change baseline. So I can't have anybody moving in, I can't have anybody moving out. I also can't have any mutations, and it's the same idea because mutations are by definition changes in alleles, changes in DNA. Well, I can't have those changes because that's going to change the gene pool. So I can't introduce any new traits. I can't remove any old traits as a result of mutations, because that will then change the frequencies in the gene pool. And the last two kind of go hand in hand. I need to have random mating, meaning that there is no preference to one individual choosing another other than you need a male and a female. And that's because any sort of preferential choice for certain genotypes is by definition sexual selection. And sexual selection is a form of natural selection, and I can't have any natural selection. That's the fifth condition here. There can't be any differential survival or reproductive success, because that will then alter the gene frequencies, and that's specifically what we're trying to avoid in this hypothetical population, because I want to see if that's actually what's happening in a real population where natural selection is likely occurring, whether it's occurring for some sort of environmental sense, or it's because it's sexual selection, and so there is preferential choice as opposed to just this random mating. So when we look at calculating Hardy-Weinberg, there are two major calculations, two major formulas that you should be familiar with. And one of them is strictly for allele frequencies. The other is there then for your genotypic frequencies. So when we look at calculating allele frequencies, we're talking P plus Q equaling 1. And what that really means is the frequency for that dominant allele, just like we were calculating back for the mouse example a few slides ago, where I have the frequency for that dominant trait. I add that together with the frequency for the recessive trait, and it ends up equaling 1. Or you can think about it, if it's in percentages, as 100%. Most of the time when we calculate Hardy-Weinberg, we leave them in that decimal form, but essentially it's the same thing. So I need those two allele frequencies to add up to 1 or 100%, because it's supposed to calculate for my entire population, and so I better include everybody. Now, if I want to look at genotypic frequencies, right, this is where I'm talking, do I have two capital letters? Do I have one of each? Do I have two lowercase? So P squared is your homozygous dominant, right? So that's where I'm now looking for the situation of being like this, right? Your heterozygotes are the frequency of being like this, where I have one of each. And then my homozygous recessives look like that, right? So when I'm up here, I'm just looking for the frequency of the capital A. Q is just the frequency for the lowercase. And then when I get into those genotypic frequencies, do I have two capitals, do I have one of each, or do I have two recessives? And we use the 2's and the squares in this situation because we're talking about diploid organisms here. So there are a handful of organisms that exist, at least in some part of their life form, as haploid organisms, meaning that they only have one allele for each trait. So Hardy-Weinberg is only going to be functional for diploid organisms, meaning that they spend the vast majority of their life cycle having two alleles for a particular characteristic. So you get one allele from mom, one allele from dad. So just something kind of as a side note to keep in mind, that this is only useful in most situations, but not everything. So let's take a look at a really simplistic example, and we'll work through some others together in class as well. So if I've got 15 individuals in my population, that means that there's a total of 30 alleles, because these are diploid organisms. So 15 times the 2 alleles they have to carry, that's where you get the 30 total alleles from. If I have 6 alleles in that population that are of the recessive or the lowercase variety, and 24 that are the dominant or the uppercase variety, then my frequencies look like this. So 6 of the recessives divided by the 30 total gets me that 0.2 frequency. 24 of the dominant divided by the 30 total gets me a 0.8 frequency. So 0.8 plus 0.2 gets me to 1, which means I've accounted for all of the alleles in this population. Now, if I look at the alleles in successive generations, I would expect, if nothing were occurring, that those frequencies don't change. If those values remain the same, then no evolution is happening. Right? There is no particular preference for that given characteristic in that given population at that time. However, if those frequencies all of a sudden become 0.4 and 0.6, right, then those values have changed, and now that means something is preferentially choosing, selecting for something about that lowercase, that recessive trait, is more adaptive in this environment given the conditions, and so more of them are the ones surviving and reproducing as opposed to those who have that dominant characteristic. And then we can start digging into what is happening, what environmental conditions might be selecting for that particular characteristic, because clearly evolution is occurring at the time.